Okay, in this presentation, we are going to talk about immunoglobulins, the structure, classification, and functions of these molecules. First, let us define what these immunoglobulins are. So they are molecules that are produced by our body to respond to foreign substances, which we know as antigens. They are also known as antibodies. These immunoglobulins are found in the serum, and if we can see here the electrophotogram, this represents the different forms of proteins in the serum. So we have albumin, alpha 1, alpha 2, beta, and then the gamma globulins. Gamma globulins is another word for immunoglobulins. General functions of immunoglobulins. Number one, they can bind with antigen, forming an antigen-antibody complex, which can result in protection. So this is what we call immune response. Valence. This is the number of antibodies that can bind with antigen molecule. Right, so when they bind with antigens, these antibodies can fix the complement, and they can also bind with other cells. Right, another property of immunoglobulins is that they are heterogeneous, meaning they can have many different forms. That is why we have different classes of antibodies. On the other hand, for myeloma, which is a kind of abnormality when the B cells produce antibodies, the kind of antibodies that they produce is only one type, right? So that means it is monoclonal. On the other hand, normally it should be polyclonal, right? So this is the structure, general structure of an immunoglobulin molecule, right? So normally we represent an immunoglobulin molecule like a letter Y. Well, what do we see in this slide? We can see that it is actually colored into yellow, red, and blue. The yellow part represents the heavy chain, while the blue part is the light chain. Aside from that, so they are connected to each other. Each of these structure is made up of carbohydrate, and these carbohydrate molecules are bound together by disulfide bonds. Within the molecule and between the molecule. Right, so there's also this part which holds these two parts together, fragments together. We call this the region, hinge region. And you can see the letters V and C. Those represent the variable region and the constant region of the molecule. So we already talked about those. Next, they have the domains. The light chain is a particular domain, so that is the inner part over here, light chain domain, and the heavy chain domain, which is made up of, again, variable and constant regions. Each of these structures are carbohydrates, which is primarily oligosaccharides. So that means that the number of carbons in these would be more than 12, because a monosaccharide may have about 6 carbon atoms in each molecule. What else do we want to say about antibodies is that we have already said this, that they are found in serum if we make a electrophoresis graphical representation, we call this electrophoregram, it will produce this structure. This is the normal amount of serum proteins. The most abundant would be albumin, and the rest would be the globulins, but they are classified based on their ability to migrate to the um, in electrophoresis, they can be classified as alpha 1 if they reach the alpha 1 level, alpha 2, beta, and then the gamma area. So those that are found in the gamma region is called the immunoglobulins. Next slide. There are 5 different classes of immunoglobulins, and again, there is a reason why we call them IgG, IgA, IgM, IgD, and IgE. We can see the same structure, although it looks a little different, but it still has basic parts, the heavy chains, the light chains, and what else? The tip of this area in the variable region is called the antigen binding site. Right, so what else? So in this area, we can see that generally the upper part, the one that forms the V, is the so-called FAB. That means fragment antigen binding. And then the lower part, the tail part, is called the fragment crystallizable. Next slide. This pertains to the heterogeneity of the antibody molecule. So that means heterogeneous, meaning they represent many different appearance. So under this, we have isotypes, allotypes, and idiotypes. The isotype is the kind of antibody that is found in the same species. An allotype is that which is found in um, related beings having the same parent or derived from the same genes. Idiotype pertains to the variable region, the difference in the variable regions of the different types of antibodies. They have these properties, complement fixation. They can fix the complement and they are able to pass through the placental barrier. Only IgG and IgM can fix the complement, while IgG is the only molecule, antibody molecule, that can pass through the placental barrier. Okay, and that part which allows it to pass through this placental barrier is the Fc, or the tail part of the antibody molecule. Right, so this is just to emphasize again, isotypes, isotypic determinants. Is this a part that is found in the different, the same species and the same species, but found in the different immunoglobulin class and subclass? Right, so when we say inbred, so that means they may come from the same parent, but they are made to produce another being. Idiotype, so that is um, the variable region that pertains to the variable regions of the antibody molecule. So each part is called an idiotope, and together we call them idiotype. So to show that explanation, we have here the difference between IgG1 molecule versus an IgM molecule from the same species, mouse. Right, so we can see that in the tail part, although they may look the same, they may look the same in the upper part, even the light chain is the same, but the heavy chain in the variable region is different. Okay, but they may differ in the tail part or the Fc region of the molecule. In the allotype, okay, so they come from the same organism, the same immunoglobulin class, but different strains. Okay, we can see here that there is a difference, a slight difference in the receptor found in the antibody molecule for the first strain A, and in strain B, it's a little different from that of the first strain. Idiotype, so again, this is the antigen, the variable region, and we can see again, so this is the same antigen, the same antibody, IgG1, but against antigen A, and this is the same antibody against another antigen. They have a different configuration. We can see here the shapes of the receptors are different, and we call them idiotopes individually. Together, that's the idiotype. Okay, another way to describe antibodies is that it's possible to separate them into two fragments. And we refer to these two fragments as the FAB and the FC. If we use an enzyme, for example, papain, this papain will act on the hinge region of the antibody molecule, cleaving it into two different fragments. The FAB meaning antigen binding, and the FC meaning fragment crystallizable. So this FC region can actually have effector functions, for example, complement fixation and placental transfer. Did I mention that? So just to emphasize the tip of this variable region, this is the variable region, okay, especially the upper area, that the variable region is the antigen binding site, and the lower part is the binding site for Fc receptors. So what are those receptors for? They can be for complement binding. They can be for complement binding, and again, they can have another function, which is to allow for transfer to through the placenta. If we use another enzyme, which is pepsin, it will result in a different um, cleavage, because this pepsin will act directly on the um, part of the heavy chain in the tail region, and it will not act directly on the hinge region. So it will result in a um, a completely intact hinge region, which attaches the FAB. We call this the FAB sub 2, FAB prime sub 2, and the remaining other fraction would be in the form of free peptides. Right, so there are 5 different classes of immunoglobulins. Immunoglobulin G, M, A, D, E. They are named based on the heavy chains that are present in the molecules. For example, gamma, the Greek letter gamma is the G. The Greek letter mu is the M. Alpha, A, delta, D, and epsilon, E. So that is the reason why they are called those designated letters. But I would say that it is more or less arbitrary. So that means there is no clear reason why they were named like that. Subclasses. Each of the immunoglobulins can have a subclass. For example, in IgG, it may have about 4 subclasses designated by gamma sub 1, gamma 2, 3, and 4. So again, it still is about the heavy chains. What else? IgA can have 2 different subclasses, A1 and alpha 1 and alpha 2. Okay, what about the light chains? The light chains can also have an effect on the subclasses of the molecule. And we have 2 different light chains, types of light chains, the kappa and the lambda chain. The human immunoglobulin light chain subtypes would have subclasses as well. So lambda 1, 2, 3, and 4. And what else? Immunoglobulins. Nomenclature, so that means the naming. How do we call the immunoglobulin? So we still use the same configuration. We use Ig, meaning immunoglobulin, the class based on the heavy chain, and it can also represent the subclass and the light chain that is present if important. It can also be simply IgG. Heterogeneity, so that means they can have many different forms. So in the summary table, we can see the types of heavy chains, the amount of antibodies that are found in serum, the sedimentation constant because they are protein molecules, they tend to be have some kind of density which is expressed in terms of Svedberg unit. The letter S then means Svedberg unit. Okay, and it has something to do with how heavy the molecules are. So knowing that IgM is the biggest molecule, it should have the highest sedimentation constant and the highest molecular weight. Okay, what about the half-life? This is the number of days they survive in the circulation. So the highest would be about 21 days for IgG1 and IgG4, okay, followed by IgG2, then IgM, and the rest would have less than 10 days of stay in the circulation. What about this distribution percent intravascular? So that means the amount more or less in terms of percentage. The number of uncirculating antibody in the blood. So about 45% or in the different forms of IgG subclasses and 80%. 80% of the IgM is found in the blood, 42% for IgG, IgA subclasses, 75% for IgT, 50%, so about half for the remaining other classes for in the circulation. And the other information we have here is the amount of carbohydrate that may be present in the molecule. So let's now go to the specific immunoglobulins. The first one is the IgG. So as I told you, this is a monomer. It has a single structure. Okay, and there are four subclasses, IgG1, 2, 3, and 4. But there is a difference between IgG3 and the rest of the other immunoglobulin Gs. It's just that the hinge region of the IgG3 is much longer compared to the other immunoglobulin subclasses, G subclasses. Characteristics, it's a light molecule, only 150,000 molecular weight in Daltons, but it has the highest serum concentration and it is capable of crossing the placenta. And this is very important in the diagnosis of certain diseases, immune diseases, because this is a antibody that increases during secondary immune response. There are four subclasses in monomer. We already said that. The next one is called an IgA. IgA is a secretory antibody. It's found in the secretions. It can be a dimer or a monomer. So if it's a dimer, it consists of these two structures. So each one is a monomer and it is joined together by the J chain. Since it's a secretory antibody, it's found in the tears, saliva, GI tract, and colostrum. It has been found to be abundant in the epithelial cells. Okay, the epithelial cells where it provides some sort of protection against enzymatic action and chemical degradation. It also provides protection against viruses and bacteria in the mucosal epithelial cells. So if you try to think about it, what are the examples of organs with these mucosal epithelial cells? So that would be the part of the digestive tract and the respiratory tract and maybe the urinary tract. So I already said that it can be a dimer and it is connected by a J chain. Next is IgM. IgM is pentamer. It has 5 arms. It has 5 arms, and it's usually the first antibody that is produced in response to initial infection, so primary response. It is unable to cross the placenta because of its big size, but it can result in agglutination. When they act against antigen, it can result in agglutination, and this is very efficient in complement fixation. Right, so this is to compare the effect of IgG, as we said, and IgM. They are the only antibodies that can activate the complement or fix the complement. Right, so if it is IgG, so this part over here will bind to the antigen. Okay, and upon binding to the antigen, it can also activate the complement, but it will not result in activation. Okay, on the other hand, if it is the IgM, it is a very efficient molecule for activating the complement, as we have said. So it will result in activation. IgE. Right, so normally we start with IgG, MAD, and then E, but we start after IgM, we talked about IgE. There is a reason for that. This IgE has an extra constant region, so that is the extra constant region, okay, which we call the C4. It is the lowest in terms of concentration in the serum, and it binds to mast cells. So if it is connected to mast cells, what could be the possible function of this IgE? So it has an effect on allergic reaction. It also provides protection against parasitic infections. And then the last one is the IgD. So it's also very low in the serum. Okay. The reason why we put it in the last is because the function of this IgD molecule is still largely unknown. Although, of course, in the future we may discover new things about this antibody, but for now what we know is that it can act as antigen receptor for B cells. Okay, because we don't really know anything about this IgD, it is not really a very useful parameter. No need to measure clinically, as it would not help the physician to make any um, wise decision. Right. The next one here would be about antigen and antibody reaction. So there are theories about how antigens and antibody interact. And one of those theories is that it folds. Okay, this so-called apple analogy theory. So that means it holds the antigen in between its arms. Okay, it forms a cleft in the V region. And then the antigen binding site is actually a small region. It is only the tip of the variable region. And this is also the antigen determinant site, which we can also call epitope. So as I have said, there are theories regarding the how antigen and antibody interact. So there is a so-called conformational, which I just described. So it fits the induced fit like an induced fit theory, or sequential. So that means there are steps that are to be followed in the um, antigen antibody reaction. Right. In any case, when antigen binds with the corresponding antibody, what will happen? Number one, it can result in agglutination of the antigen. It can initiate an immune response, which can be cellular. It can activate the macrophages, the phagocytes, or even the natural killer cells. And humoral, so that may mean the production of hormones, or rather production of um, antibodies that can act against the antigen. It can also activate the complement, and it can allow for direct killing of the cells by phagocytes. So that is the last slide we have for antibodies. I hope it's clear, and if it's not clear, you can always go back and check the different slides we have for this presentation. Thank you for listening.